Good morning all. Ready for the class? Today we will discuss about the product synthesized from glycine. In the last class, we have learned about the different types of amino acids, importance of amino acids, etc. So today we will go to the product synthesized from the amino acid glycine. So amino acids are a group of organic compounds containing two functional groups, amino and carboxyl. The amino group is basic and while the carboxyl group is acidic in nature, which we have already discussed in the last class. So glycine is a non-essential, optically inactive and glycogenic amino acid. What is glycogenic amino acid? Any idea? It is precursor to glucose. Yes. It is indispensable for chicks. Why it is indispensable for chicks? The glycine plays an important role in metabolic regulation, antioxidative reactions and neurological functions. So this glycine is one among the commonest amino acid found in protein structure. Glycine is also an inhibitory neurotransmitter in central nervous system. It is the most abundant amino acid normally excreted into urine. This glycine has therapeutic uses. In recent years, glycine is being used to protect against heart diseases, liver damage, improvement of insulin action, induction of sleep, treatment of schizophrenia, stroke, etc. Some of these therapeutic uses of glycine are at experimental stages. And this glycine is synthesized from serine by the enzyme serine hydroxymethyl transferase, which is dependent on tetrahydrofolate. And this glycine is also be obtained from threonine, and this reaction is catalyzed by threonine aldolase. And glycine is actively involved in the synthesis of many specialized products like heme, purine, creatine, etc. in the body, and that is the topic today. So we will discuss about the products synthesized from the amino acid glycine, which includes synthesis of purine ring, synthesis of glutathione, conjugation reaction, biosynthesis of heme and biosynthesis of creatine. First we will discuss about the synthesis of purine ring. Here the entire molecule of glycine is utilized for the formation of positions 4 and 5 of carbon and positions 7 of nitrogen of purine. You can see here in the red color. Next one is synthesis of glutathione. It is a tripeptide. Glutathione is the most abundant non-protein thiol in the cell. It is found in two forms. Okay, one is glutathione and the another one is glutathione disulfide. And this glutathione is often called reduced glutathione. And the glutathione disulfide is often called oxidized glutathione. And glutathione is synthesized in 2 ATP dependent steps. Here you can see the synthesis of glutathione. First, gamma glutamyl cysteine is synthesized from L-glutamate and cysteine via the enzyme gamma glutamyl cysteine synthetase. This reaction is the rate limiting step in the glutathione synthesis. And second one is glycine is added to the C-terminal of gamma glutamyl cysteine via the enzyme glutathione synthetase. Next one is conjugation reaction. As a conjugating agent, glycine performs two important functions. Okay. The bile acids are conjugated with the glycine. What are the bile acids? Cholic acid and chenodeoxycholic acid. See, the cholic acid conjugated with glycine gives rise to glycocholic acid. And chenodeoxycholic acid conjugated with glycine and gives rise glycochenodeoxycholic acid. And what they do? They convert a weak acid to strong acid. As a result, The conjugated bile acids are fully ionized at the range of pH values present in the small intestine. And glycine is important for detoxification of benzoic acid to hippuric acid. Next one is synthesis of heme. Glycine condenses with the succinyl-CoA to form D-aminolevulinic acid, which serves as a precursor for heme synthesis. And this reaction is catalyzed by the enzyme ALA synthase, which is a rate limiting enzyme in the heme biosynthetic pathway. Next one is biosynthesis of creatine. Creatine is present in the tissues like muscles, brain, blood, etc. It is present as a high energy compound, phosphocreatine, and as a free creatine. Glycine, arginine, and methionine are required for the creatine formation. Creatine is formed by the spontaneous utilization of creatine or creatine phosphate, and this creatinine is an anhydride of creatine. Creatinine is excreted in urine. So here you can see the biosynthetic pathway of creatine. The first reaction occurs in the kidney. It involves the transfer of gonidino group of arginine to glycine. This reaction is catalyzed by arginine glycine transaminidase to produce gonidoacetate. Okay. Then S-adenosyl methionine, that is active methionine, donates methyl group to glycosamine to produce creatine. And this reaction occurs in liver. Okay. The first reaction occurs in the kidney. Okay. And this reaction occurs in liver. Creatine is reversibly phosphorylated to phosphocreatine by creatine kinase, and it is stored in muscle as high energy phosphate. Okay. That's all about the product synthesized from glycine. Everybody understood? Any doubts? If you have any doubts, you can ask. Thank you.